Friend, today I will discuss about the chapter sexual reproduction in flowering plants. In this chapter, I will discuss about microsporogenesis. Last lecture, I have told you about parts of flowers. Now we are seeing here how microsporogenesis is performed. So, what is microsporogenesis? Microsporogenesis is a process of formation of pollen grains inside the pollen sac of flowering plants by meiotic red or reduction division. Each microspores is the first cell of the male gametophyte that produces the male gametes. I think the summary of the microsporogenesis have you understood? Let us see how do you understand this? How will you understand this? So I saw the contents, the steps which were come to the formation of microsporogenesis. A process in the process of microsporogenesis. The first step, pollen sac, formation of pollen sac, then pollen grain, and then male gametophyte or germination of. Now I give you dramatic explanation of this microsporogenesis. So you see, if the mature suppose anther and filament, this is the male part of the flower. Now see in this picture A, this is the anther. When you cut the section, it is the purpose in this diagram. The purpose is so how pollen sac or anther, how is microspores and how microsporangium is formed. So say if you TS section, cut the TS section, in this picture you see the 4 lobes, 1, 2, 3, 4 lobes are seen. In each lobes, first cell is archaeosporious cell. Archaeosporious cells, and here suppose see here lobes, and first cell is archaeosporial cell. It is capacity to division. The divide, after divides antikinal divisions, it forms a large number of cells, and these cells are divided into different tissues. And what is the name? Here it's clear. The vascular bundles here, then tapetum cell, again endothecium, again mother cells, microspore mother cells, endothecium. These are the cells which are formed by a single archaeosporial cells. Then in the next, here is the overall the pictures how the pollen sac, microsporium, and male gametophytes are formed. Here see the main parts of the this one is the TS of anther, TS of pollen sac. Then you see the chamber. This is chamber, this is chamber, pore chamber, and in this chamber loculus or microspores are called microsporocytes are formed. This diploid nucleus, it has diploid microsporocytes had diploid nucleus. Then it forms haploid and callose haploid nucleus after meiotic divisions. Then meiotic division second forms 4 tetrads, that is called tetrads, and microspores are these two microspores. This 4 is formed microspores after meiotic division, and then microspores are goes and grow into the pollen grain. This is pollen grain, and pollen grain has vegetative nucleus and generative cells. Both bodies are found in the pollen grain, and outer layer is pollen grain. Outer layer is hard exine intine cords, and then pollen grains are grow into fashion with meiotic divisions. One second mature pollen grains are divided, means it has a germ pores, and this germ pores from this germ pores the pollen generative nucleus divided in two parts. Vegetative cells and generative cells are divided in two parts. Vegetative cells nucleus, generative nucleus is vegetative nucleus developed the pollen tubes, and these pollen tubes are entered into the pistil. Next figure, it is more clear how to develop male gametophyte. Just see the microsporocyst, then meiosis first 2 nucleus divided in 2 nucleus, then meiosis second 4 nucleus. This is haploid nuclei, and then this haploid nucleate is formed microspores, that is tetrads, because it is the number of 4, and next is the released microspores, and then microspores have vacuoles and vegetative nucleus, and then vegetative nucleus. In this picture, bicellular pollen means bicellular pollens are formed. Generative cells and vegetative cells. That generative cells are developed into tricellular pollen. Pollen means 1 vegetative nucleus and 2 sperm cells. Now, this is the formation of male gametophytes. Now you can see in this picture the details of the pollen grain, structure of pollen grain. The outer side you see the germ pores, vegetative nucleus inside and vegetative cells, and next cytoplasms. Intine, inner layer is called intine, or outer layer is called exine, and one is generative cells. This generative cells, here the most clear, more clear than how male gametophyte is formed. This is the pollen grain. Pollen grains, you see the large vegetative cells, small generative cells, and smaller generative cells, then vegetative cells nucleus, and meiotic division. Generative cells are divided in two parts, and then two male gametes inside when pollen tubes are developed. Clearly you can see the pollen male gametophytes has two male gametes, means, and one pollen tube. It has tube nucleus. It was developed on the stigma of the female parts, that is the pollen grains and microsporogenesis process. So students, you see the diagram. I my best try to you to explain the diametrically how to pollen sac, how to pollen grains and female gametophytes are developed. In this, one thing I want to say, endothecium and tapetum, both cells are found inside the pollen chamber, inside the pollen sac. And this is the notes for you. You please note down the notes about the microsporogenesis, and you use the diagram at the particular places which help you to discuss about the microsporogenesis, and easily will understand these points. Now I think you have all about understand the microsporogenesis. If any doubts, please consult your subject teacher, and if it is not clear for you, so please comments on my YouTube channel comment box. I will certainly give you answer. Thank you. Best of luck.